the Vault story to a very broad audience. And I understand that I'll be speaking with undergraduates to graduate students, as well as cell biologists, chemists, and engineers. I invite any students and non-cell biologists who might have questions about my talk today to visit my YouTube lecture series, which is called Lessons with Lenny. My goal in this series is to explain vaults and teach basic cell biology and nanotechnology. For my colleagues who are tuning in today and want more details, I have included some key literature links at the bottom of some of my slides. This literature link here, let's see if I get my laser pointer to work. This literature link just happens to be the nicest article that anyone has ever written about me and vaults that came out last June in Science magazine. Shout out to the Science News editor, John Travis. So today I have three major goals. First, I'm going to describe the vault history. Second, I'll give you two examples of vault engineering. And finally, I'll mention some work on attempts to determine vault function. So let's begin with some vault history, which actually vault history starts with Rome history. I was born in Youngstown, Ohio. My science life started at the University of Michigan, where I earned a Bachelor of Science in Chemistry and a Ph.D. in the lab of William E.M. Lands at the University of Michigan Medical School in the Department of Biological Chemistry. After my Ph.D., I spent nearly 5 years at the National Institutes of Health, which is in Bethesda, Maryland, at the laboratory of Elizabeth Neufeld, where I studied cell biology of an organelle called a lysosome. Believe it or not, that's me back when I was a postdoc. In 1979, I moved to Los Angeles, California, to accept a faculty position at UCLA in the medical school in the Department of Biological Chemistry. And I liked UCLA so much that I have never left. During my time at UCLA, I was also for 12 years combining my research life with an administration role while I served as the research dean of the medical school. After a couple years at UCLA, I had established my lab and attracted a graduate student, a postdoc, and hired my first technician. Here's my group in the early 1980s. That's me on the right there. My lab studied lysosome biogenesis and focused on how lysosomal enzymes got transported through the cell to the lysosome. Here's a cartoon showing the movement of lysosomal enzymes to the lysosome. We concentrated on these little vesicles here, which are called coated vesicles. And my first graduate student, Corinne Campbell, purified these vesicles and demonstrated that they contained lysosomal enzymes. Here's negative stain of these coated vesicles. In 1984, Nancy Kadersha joined my lab as a postdoc, and Nancy developed a new procedure to purify coated vesicles using agarose gels. The coated vesicles that she prepared were very pure. However, there was a fraction of coated vesicles that were contaminated with a smaller ovoid structure. Here's the coated vesicles, and here's the fraction that was contaminated with this structure. And when we published a paper on the fractionation of coated vesicles, we referred to these structures as a novel ovoid particle. So Nancy purified the particle because we thought it might be some kind of coated vesicle. But after several months of study, we realized that the particle was not at all related to coated vesicles, and therefore it was something new. We had so many questions. What was their structure? How common were they? Why hadn't they been seen before? And most importantly, what should we call them? So we organized a Name That Particle competition in the lab with everyone participating. And there were lots of suggestions for the name that we should put forward, including hot dogs, raspberries, even sushi rolls. However, I don't think they look like sushi. Maybe a little bit. We finally went on with one of Nancy's suggestions, which came from the particle's shape. The images displayed a very distinct and complex shape resembling the arched ceilings that form vaulted cathedrals, ceilings of cathedrals, and therefore we adopted the name vaults. When we measured the mass of the particle, it turned out that it was 13 million Daltons. This is about one of the largest naturally occurring cell particles ever discovered. Here's an SDS page gel of pure human vaults, showing the protein composition of the particle and the identification of all the major components which occurred over the next 10 years. Vaults are enormous, yet they're only composed of 3 protein species, shown here on the Kamasi gel, and a small untranslated RNA, shown by this dotted line that doesn't stain with Kamasi. The major structural protein of the particle is approximately 100 kilodaltons that we called the major vault protein, or MVP for short. There are 2 high molecular weight proteins found inside the vault particle. One is 193 kilodaltons. The other is 280 kilodaltons. We name the 193 kilodalton protein VPARP, or Vault PARP. This protein turned out to be a member of a family of enzymes called PARPs, for poly-ADP ribosyl polymerases. This Vault PARP is now called PARP4. The 280 kilodalton protein was originally found and named because it was in the telomerase complex, and it was called TEP1 for telomerase protein 1. TEP1 is an RNA binding protein. This red domain is where RNA binds to this protein. And it originally was thought to bind the telomerase RNA, but when it's in the vault particle, it binds a small RNA. In humans, there are a couple of highly related vault RNAs ranging from 86 to 89 bases, and that's shown here on the gel. It turns out that vaults are highly expressed in almost all higher eukaryotic cells. Vaults are found in the cytoplasm of cells, and the average cell of the human and mouse contains about 10,000 vault particles. Some cells have over 100,000 particles. Here's a human skin cell called a fibroblast, stained with an antibody against the major vault protein. And as I will describe to you in a few minutes, all of this staining is due to vault particles. The smallest dots here on the immunofluorescence are single vault particles. After we cloned the first major vault protein gene, we could search sequenced genomes, and we found that vaults were present in nearly all eukaryotes, but not in prokaryotes. However, to date, vaults are missing in certain notable eukaryotes. They are not found in yeast, nematodes, most insects, and they're not found in plants. A careful genetic study by Tony Daly proposed that vaults were present in the last common eukaryotic ancestor, LECA. LECA is a hypothetical ancestor of all living eukaryotes, and thus it appears that vaults are ancient, arising nearly 2 billion years ago. We examined the 3D structure of vaults using cryo-electron microscopy and image reconstruction with Phoebe Stewart originally, and our low-resolution structures revealed that the particle is bifold symmetric capsule. The cryo-EM structure was hollow inside. This apparently is because the internal components are mobile. And like in crystallography, if something is moving around, it's not imaged in cryo. However, if you look at a density slice through the particle, you can see that there is density inside. And the density is made up of the internal vault components that I described. TEP1, I should mention that the major vault protein makes up the entire shell of the particle, but the TEP1 protein and the vRNA are up near the cap of the particle, and since the particle is bifold symmetric, you get TEP1 and vRNA up near the top and the bottom, and the V-part protein is this density near the center of the vault, shown here in pink. The precise size of the particle was determined by cryo-EM to be about 70 nanometers by 42 nanometers. My postdoc, Andy Stephen, wanted to study MVP as a protein, so he expressed it in insect cells using a protein expression system called the baculovirus expression system. And in this system, you start by inserting the MVP cDNA into an insect virus called a BACMID. What Andy found when he did this was very interesting and very confusing at first. And what he found was that he could not find the cells making any free major vault protein. But to our surprise, they did assemble and make perfectly nice-looking vaults. These vaults appeared to be structurally identical to naturally occurring vaults, except they were hollow inside because insect cells do not have any of the other vault components, the TEP1, the VPARP, or the RNA. We decided to try to map the orientation of the major vault protein in the particle. And to do this, we constructed a major vault protein with 33 extra amino acids attached to the N-terminus of the protein, illustrated by this pink squiggle here. And when we generated a cryostructure, the outside of the particle looked exactly like it had when there was no tag added to the MVP, but we were able to locate the tag by carrying out what's called a difference map, essentially subtracting the vault with no tag from the vault with a tag. And the difference density showed the only difference at the waist of the particle on the inside. I've taken a slice through the particle here and flipped it on end, and this pink density that shows up here is the difference density, which we think was the tag. In other words, this is the only difference when we add these extra amino acids. We also made a vault from a major vault protein that had extra 12 amino acids at the C-terminus of the major vault protein. And when we carried out a cryo-reconstruction of this vault, we didn't need to do a difference map because the difference density was an extra blob of density at the top and bottom of the particle. And this led us to a model that predicted that the major vault protein ran from the top of the vault at the C-terminus to the waist of the vault at the N terminus. And this was similar to our prediction. The resolution of cryo at that time was not very great, so we turned to X-ray crystallography to try to get additional support for the model, and partnered with David Eisenberg's lab at UCLA, and we were able to get a 9 angstrom structure. It took David's lab, Dan Eisenberg, 3 years to grow a crystal of the vault that diffracted at 9 angstroms, but shortly thereafter, Hideki Tanaka in Japan, who spent 5 years growing a crystal of rodent vaults, published a 3.5 angstrom structure for the particle that's shown here. I've colored one MVP chain red so you can see how it is aligned in the particle, and sure enough, it aligned as we predicted from our cryo structure. There was a surprising finding from Hideki's structure, and that was that Hideki showed that there were 78 copies of MVP in each vault particle, or 39 copies in each identical half vault. And that was kind of confusing at the time. I didn't understand how nature could work in multiples of 39. And that really came out of our model at the time for vault assembly. We thought that vaults assembled like viruses, that they self-assembled. And the model at the time was that 39 MVP proteins came together to form half vaults, and the 2 half vaults came together to form the whole vault structure. There was other data, though, that was not consistent with this model. First of all, when we expressed the major vault protein in cells, like I just told you, we never found free MVP chains produced by the cells, only assembled vaults. In addition, when we took vaults apart into individual MVP protein chains, we couldn't get them to reassemble again, no matter how hard we tried, and we tried very hard. Finally, we did a series of experiments that completely killed our self-assembly model, that we knew this model was incorrect. We thought these experiments were based on a thought that we could make vaults from a major vault protein that had a very large number of positively charged amino acids added to the N terminus of the major vault protein, and therefore, when the half vaults formed, they would have these charged residues at the waist, and that would prevent the 2 half vaults from coming together by charge repulsion. So my postdoc, Jan Mrazek, fused 51 amino acid human protein called protamine to the MVP N terminus. Protamine is a small protein important for packaging DNA into the head of a sperm cell. And 24 of the 51 residues of protamine are the positively charged amino acid arginine. So this would give each MVP chain an extra 24 positively charged residues. However, when we made this vault and looked at it, it was quite puzzling. Instead of making half vaults, the cells made perfect whole vaults. These vaults appeared to be the most perfect looking vaults we had ever seen. There were no broken or half vaults present, and these vaults were even more stable than vaults without the protamine tag. Around this time, we had started working with a new cryo-EM group at UCLA led by Hong Zhao. Hong's group carried out a cryo-EM reconstruction on the protamine vault that Jan had made. And here is the structure, a cut view of the structure. And we can see the protamine chains forming a tight donut-like cluster at the waist of the particle. This is a pseudo-colored cryo-EM structure, and the material clustered at the waist are the 78 protamine chains colored yellow and orange. These would be highly charged. And when we modeled a rough protamine structure within this extra charged area, we could see that the protamine chains were crossing each other and forming this highly charged packing against one another at the waist of the particle. And so the formation of this vault was completely inconsistent with our self-assembly model. How could two half vaults come together, each terminating in nearly 1,000 positive charges, and overcome the significant charge repulsion that would be required to come together? This was just inconsistent with half vault formation. So we started to examine the Tanaka high resolution structure of the vault, which is again shown here. And if we zoom in on the contacts between the N-terminal chains coming from the top half of the vault, colored green here, and the bottom of the vault, colored blue here, we see that most of the important contacts at the waist are at the first few amino acids of the major vault protein N-terminus. And if we zero in on these 10 amino acids, we see them here, starting with the methionine. And Jan decided to probe the vault structure by making substitutions in these actual amino acids with the amino acid histidine. Histidine is a small neutral amino acid that is often used to probe protein structure. And he started to replace the third amino acid, which happens to be a threonine here, with a histidine. And what he found was that the vaults were pretty much normal looking when he did that. And so he continued to replace amino acids with histidine, with 2 histidines, 3 histidines, 4 histidines, and even 5 histidines. He continued to get structures that looked like vaults and whole vault particles. But when he put 6 histidines into this structure, he saw no vaults whatsoever. Instead, he saw these very large structures that looked like rolls of aggregated protein. Occasionally, these rolls looked like they were unraveling. Jan used our antibody that was made against MVP to demonstrate that these rolls were made from MVP. When we examined the rolls at high magnification, they appeared to roughly align with the structure of the vault. The center of the large parts of the roll were aligned with the center, and the shoulder region and the cap. When we looked at the unraveled rolls at high magnification, these made us think that they were made from dimers of the major vault protein. If you put a dimer together, it forms a structure like this. So we were thinking that maybe these were dimers. And that got us thinking more seriously about protein transcription and translation. And we took a deep dive into understanding protein synthesis, which takes place on structures called polysomes. Polysomes are very difficult to visualize. However, we uncovered a classic paper published in 1982 that showed a giant polysome from a small fly called a midge. These polysomes are massive since they're translating proteins that are 5 to 10 times the normal size of proteins. And let me explain what's going on in this interesting micrograph. We're looking at a polysome chain, and the black blobs here are ribosomes. And so I will label the ribosomes green so you can see them. And these ribosomes are moving along a chain of mRNA that's too thin to stain with negative stain. And instead, I've colored in the mRNA chain orange, labeling the beginning of the chain the 5' end and the end of the chain the 3' end. The ribosomes are moving from the 3' to the 5' end and producing the growing protein chain, which I'm coloring here red. And you can see as the chains are moving, are getting longer and longer, as the ribosome is moving to the end of the mRNA, the protein chains coming off the mRNA get longer and longer. This turns out to be a wonderful way to think about how proteins are made. So we isolated polysomes from our insect cells that were making normal vaults, and they weren't very informative. However, and you can see them here. They're much shorter because these are shorter proteins than are being made in the midge that I showed you a minute ago. But what got interesting was when we isolated polysomes from the insect cells that were making the 6 his tagged vaults. And then what we saw was these large structures were attached to the end of the polysomes. And this really led us to finally understand vault assembly. And it explained all of our data, and it explained why we never saw free major vault protein in the cell. And the answer is because the cell does not make free protein. It assembles vaults, one vault at a time, on the polyribosome. And this was published in ACS Nano and featured on the cover, but it can best be explained by a brief animation, which I'll show you now. The messenger RNA that encodes the major vault protein MVP is depicted by this thin orange line. Ribosomes are the cellular machines that translate the message into protein. The large and small ribosomal subunits, colored blue and yellow, attach to the mRNA and move along and produce the growing MVP chain, illustrated in red, emerging from the ribosome exit chain. Successive ribosomes attach and proceed along the mRNA. This arrangement of multiple ribosomes decorating an mRNA chain is called a polyribosome. Within a few seconds after protein synthesis begins, we can see the newly formed MVP chain emerge from the first ribosome. When about half of the MVP chain is produced, it's long enough to make contact with the growing MVP chain emerging from the ribosome, moving along just behind it, forming a dimer. Behind the first dimer, there's another dimer that is formed. And beyond that dimer, yet another dimer forms. If you focus your attention on the right side of the polyribosome, you can see what happens as the first MVP chain is completed and the first ribosome falls off the mRNA. The MVP dimer stays tethered to the polyribosome as the ribosomes leave. The first MVP dimer forms side-side contacts with the second dimer. It's coming up behind. What is seen as a growing sheet of MVP dimers that remain attached to the polyribosome as more and more dimers join. The MVP dimers curve up away from the polyribosome and begin to form the body of the vault particle. The final vault structure is realized when 39 MVP dimers come together and the finished vault pinches off from the polyribosome, and the final folding process of the vault continues to completion. So this model explains why we didn't see MVP chains, only vaults made, and it explained all of our other data. And especially it explains why the vault structure is so regular, because each vault is made the same way, and therefore the vaults were regular enough to do X-ray crystallography and crystallize them. So now that we've covered the basics of vaults as they exist in cells, I want to give you a look at why and how we engineer the particle as a flexible nanoscale biologically interesting container. I'll cover the strategy we developed for packaging cargo into the vault, and then I'll go into two applications in a little bit more detail. So how are we going to package material into the inside of the vault particle? And this is a good time to introduce you to Valerie Kickofer. Valerie joined my lab as a postdoc in 1989, and she stayed in my lab as a research associate until she retired a couple of years ago. In order to package cargo in the vault, we borrowed some information from the VPARP protein. Remember, VPARP is packaged near the waist of the particle. Valerie had identified the VPARP protein using a screening method called yeast-2 hybrid system, and she figured out that VPARP was packaged into the vault particle through an interaction between the C-terminus of VPARP and the major vault protein. And this domain, this interaction domain, which we called the major vault protein interaction domain, or INT for short, serves as like a zip code to direct the protein into the inside of the vault. And we knew that this domain bound to the inside of the vault at this location on the major vault protein. So as a proof of principle, we engineered a fusion between the VPARP INT domain and an enzyme isolated from fireflies called luciferase. It's the enzyme that gives the firefly its glow. We produced a fusion protein of luciferase and the INT domain, and we produced empty vaults and mixed them with the INT-tagged luciferase fusion protein. And when we examined the vaults by cryo-EM, they looked perfectly normal from the outside, but when we did a difference map, it revealed all of the difference density. The fusion protein was in 2 rings of density in the inside of the particle. In addition, we found that these particles still had the luciferase enzyme activity, and we added the substrates to this vault. They glowed. Therefore, we had succeeded in making little nano light bulbs. So today I want to describe 2 applications that use this packaging method. One is in cancer and one is in environmental remediation. Let's start with some really basic cancer cell biology. Cancer cells arise in people like you and me all the time due to different genetic mutations. These gene mutations can be inherited. They can develop as we get older. They can be caused by environmental damages like cigarette smoke or UV radiation from the sun. I live in Los Angeles, and there's lots of UV radiation from the sunlight here. The vast majority, though, of these cancer cells that arise in our bodies are destroyed by the immune system. But rarely, sometimes, the cancer cells avoid the immune system and develop into a tumor. If I got a tumor in my lung 10 years ago, my treatment options would be quite limited. They would be surgery, radiation therapy, and possibly chemotherapy. And as you might know, chemotherapy is the use of drugs that are really just poisons that are employed under a very simple strategy. The poison kills rapidly dividing cells like cancer cells, more rapidly than it kills normal cells, non-cancer cells. But these drugs might kill the tumor, but they have significant side effects. For example, hair cells grow rapidly, and you can lose your hair from these chemotherapy, and I really can't afford to lose any more of my hair. So let's go back to immunotherapy. It's necessary because the tumors create an environment that blocks the immune cells from recognizing the tumor. And there are many different ways that the tumor cells overcome the immune system, but a field called immunotherapy has been developed to overcome these barriers set up by the tumors. And these barriers are overcome by different strategies. The first strategy that I'll talk about is called CAR-T. CAR-T is a way to educate T cells in vitro, meaning in the laboratory. This is a huge therapy now. It has a global market value of $6 to $8 billion last year. But an even bigger strategy for overcoming the cloaking that the tumor does to prevent the immune system is called checkpoint signals. And therefore, a whole group of drugs have been developed called checkpoint inhibitors. And by the way, the global market for checkpoint inhibitors last year was over $60 billion. My lab started working in this area about 15 years ago when I learned about a promising strategy to try to overcome the barrier that was proposed by the head of pulmonary oncology at UCLA, a doctor named Steve Dubinet. And Steve was asking the question, what if we could make the tumor environment more like a lymph node? And let me explain to non-immunologists what Steve meant by this. Lymph nodes are small glands throughout the body that are part of the lymph system, which carries fluid called lymph between body tissues and the bloodstream. The lymph system is an important part of the immune system. The lymph nodes filter lymph fluid as it flows through them, and it traps bacteria, viruses, and other foreign substances. And these substances are held in the lymph node so the immune system can recognize them. And the way this works is through small chemical attractants called chemokines. And the chemokine that Steve was working on is called CCL21. The chemokine sets up a chemical gradient, and this chemical gradient attracts cells of the immune system into the lymph node where they can fight the foreign objects that are trapped there, like a bacteria or a virus. So back to Dr. Dubinet. Steve reasoned that if he could deliver CCL21 into the tumor, then maybe the tumor would act like a lymph node and attract the immune system into the tumor. But this was problematic. Delivering CCL21 is a problem because it's a small protein and it could be degraded or dissipate from the tumor. So Steve had to engineer cells from his patients to solve this problem. And what he did was he isolated cells called dendritic cells from the blood of his very sick lung cancer patients. These cells are also called antigen-presenting cells, or APCs. And in the laboratory, Steve infected these cells with a virus that contained a DNA that encodes the CCL21 protein. And effectively, this teaches the dendritic cells to make CCL21 protein. And so he grew up lots of these cells in the lab, And he started by injecting these cells into lung tumors in a mouse model. And when he injected the CCL21 dendritic cells into a tumor model in the mouse, he found that it led to the destruction of the tumor. You see the tumor now being destroyed. And this was early data that allowed him to eventually try this therapy in humans. And he carried out a small safety study a few years ago where he showed that the therapy was promising in that the immune system was activated. There was a dramatic shrinking of some of these large tumors in patients, and some of the small number of patients that were treated showed dramatic increases in survival. However, there were many problems that need to be overcome before this could be a real therapy. First of all, the cost is very high. This therapy cost over $150,000 per patient. Second, it takes weeks to prepare the cells. And finally, these types of cell-based systems can be very inconsistent. So Steve needed a way to avoid the cell engineering. And perhaps he could use a nanoparticle to deliver CCL21. And what he really needed was a nanoparticle that would not trigger an immune response. And so we asked the question, maybe vaults could be used to substitute for the engineered dendritic cells. If we could make a vault particle to deliver CCL21 to a tumor, then we could obviate the need for a virus-transfected cell. Your vaults and my vaults are identical, so the CCL21 vaults could be stored and used when needed. It wouldn't take any time to make the cells. They would be an off-the-shelf reagent. You wouldn't have to wait for therapy. There would be a terrific economy of scale, streamlined pharmaceutical production and standardization, and they could be consistent and dose could be controlled. These things would lead to a much reduced treatment cost. So we set out to do this and make this vault particle. And here is the CCL21 vault that we made. We did this by fusing the zip code I just told you about, the int domain, to the CCL21 chemokine. You can see on a gel that the vaults, the purified vaults, now contain the chemokine. And interestingly, when you look at these vaults in the electron microscope, you actually see two light regions where the CCL21 that's packaged inside the vault causes a bulge in the vault that shows up as these white bands inside the vault when they're looked at in negative stain. So does the CCL21 vault work to shrink tumors? And this graph shows a growth of a tumor in a mouse model that was treated with an empty vault, which is the same curve that you get if there is no treatment at all. But if we use a CCL21 vault, we see that the tumor is dramatically slowed in its growth. And if we look at the tumor itself and compare the tumor environment with an empty vault and a CCL21 vault, we see that the CCL21 vault has attracted a large influx of immune cells, these small green cells that you see in the tumor. They have been attracted to the tumor, and they have stimulated a T cell proliferation and a cytotoxic T lymphocyte, or CTL, attack on the tumor. And this is precisely the mechanism that we were looking for, and we hope that this will be the first application of a vault delivery system that we can bring to a human clinical trial. So let me just give you a very brief update on where we are at taking the CCL21 vault into a clinical trial. This summarizes the development of this therapeutic from the discovery of vaults in 1986, the characterization that occurred over the next 15 years or so, the basic structure being worked out, and then in 2005, our collaboration with Steve Dubonnet. We continued to develop the Vault platform, and in 2013, I partnered with an entrepreneur to form a company called Vault Pharma with the goal of commercializing the Vault platform. And we raised money to pay for the clinical development costs, which are very high. And we developed a robust clinical manufacturing of the Vault particle and completed what's called a GLP production of the CCL21 Vault. This year, we plan to complete GMP production and up to the FDA standards, and we hope that by next year, we'll enter a clinical trial with the CCL21 Vault. So that summarizes the work on cancer, but I also want to mention today an area of engineering vaults for environmental remediation. And this work was developed in collaboration with Professor Shali Mendra, a very talented environmental engineer here at UCLA, and her former graduate student, Meng Wang. Meng is currently an assistant professor at the University of Pittsburgh. A major focus of the Mahendra Lab is biological water treatment. A major contaminant of drinking water is bisphenol A, or BPA. BPA is a chemical commonly used in plastics, paper, food packaging industries. And a large number of studies have raised concern about BPA exposure because BPA has a variety of significant adverse health effects, including and most notably a strong impact on reproduction and fertility. So, the removal of water contaminants such as BPA has been looked at for a long time, and it can be achieved with a variety of procedures, including physical, thermal, chemical, and biological transformations. But there's been a large interest in biological transformations because it can often be carried out at a lower cost and lower energy expenditure than other technologies. And biological transformations are carried out through microbes or through enzymes themselves. Microbes take a long time to degrade contaminants, sometimes months or years, where enzymes can act very quickly. There's a significant public concern about using microbes, which are often pathogens, and people don't want to release these microbes into the environment to clean up environmental toxins. But these environmental concerns are not relevant to enzymes. However, enzyme stability is limited in contaminated water. So the field is very interested in enzyme stability, which can be done in a number of different ways. You can stabilize enzymes by immobilization, such as absorption, covalent bonding, and physical trapping. However, immobilization often deactivates enzymes and denatures them, and it also can limit substrate diffusion and prevent homogeneous catalysis. So we asked the question with the Mahendra Lab, can enzyme immobilization in a Vault particle overcome some of these limitations? Several oxidative enzymes, such as manganese peroxidase and lactase, have been demonstrated to mediate the transformation of BPA by coupling reactions or scission reactions, and we decided to initially focus on manganese peroxidase, which has been isolated from a fungus called white rot fungus and shown to degrade BPA. So we expressed the recombinant manganese peroxidase fused to the int packaging domain using the same protocol that I talked to you about previously. And we incubated the fusion protein with recombinant vaults, resulted in packaging of the enzyme into the vaults. And you can see the purified vaults here and a Western for the enzyme manganese peroxidase here, and the beautiful vault particles that formed with the manganese peroxidase packaged in the inside. And when we examined the stability of the vault packaged manganese peroxidase and compared it to the stability of the recombinant enzyme and to native manganese peroxidase that we isolated from white rot fungus, we saw that there was an amazing stabilization of the enzyme by packaging it into the vault particle. The recombinant enzyme lost all of its activity at 25 degrees centigrade in 50 hours. The native manganese peroxidase was a little bit better, but by packaging the enzyme in the vaults, we got over 60% of the enzyme stable over the same time period. This extended to thermal stability and pH optimization. Here's degradation of BPA mediated by the vault packaged MNP, which was very rapid, whereas the free enzyme and the recombinant enzyme did not do a very good job in the same amount of time. In addition, the vault packaged enzyme shown here in blue had a larger pH optimum, so it could operate at pHs above the optimum for this enzyme. We also compared the effectiveness of various MNP analogs, and they were also degraded by the vault packaged enzyme. Our studies extended to looking at the cause on reproductive organisms, reproductive toxicity, and we used a C. elegans model for toxic effects of BPA and BPA analogs on C. elegans development and embryos. And we showed that the pre-treatment of C. elegans embryos with the Vault MNP dramatically reduced germline apoptosis and survival. And that's, again, these blue lines showing the Vault difference from the free enzyme. So we think that vaults have a great potential for assisting in bioremediation. They can be made by a process that's safe for humans and the environment. They enhance the stability and the pH optimum of the enzymes and improve the biotransformation rates of the reaction. However, there are two obvious barriers to using this technology for environmental remediation. And the first is the scale up. We need to be able to produce vaults at very high production levels in order to use them for environmental applications. And secondly, even though the vault is protecting the enzyme that's packaged inside, let's face it, to a soil organism, the vault particle itself would just be a tasty protein hors d'oeuvre. So how are we going to protect the vault from the soil organisms? And so I want to explain how we solve these problems. So to remind you, our CCL21 vault that I just described was produced in the baculovirus system, and we had developed the manufacturing with a manufacturing partner that was doing this in FDA-approved system. But this system is very expensive, and it only produces drug quantities of the material. And so to get around this, Meng turned to yeast, and Meng developed a system to produce vault particles in yeast using the yeast Pichia pastoralis. And recall, yeast do not have vaults. And so Meng developed a yeast strain that constitutively expresses MVP, and thus they constitutively make empty vaults. So to make a vault particle packaged with an enzyme cargo, we simply express the enzyme infusion protein in this constitutive strain, and then grow up the yeast and isolate the vaults from these yeast, and they are fully packaged with the enzyme that has been attached to the targeting domain. And we've already converted our clinical system to this yeast manufacturing system, and we're already manufacturing the CCL21 vault in fermenters, and we're moving now into 100-liter fermenters to make the vaults. And this process is very scalable, and we anticipate that we can produce kilogram quantities of vaults using fermenters and this procedure. The second problem I mentioned is protecting the vault particle, and we've done this by encapsulating the vaults in sol gels. And to our surprise, incubation of vaults at pH 6 with prehydrolyzed tetramethyl orthosilicate resulted in the vault-specific sol gel nucleation and the formation of relatively homogeneous particles, approximately 400 nanometers in diameter, formed quantitatively entrapping the vaults. When we examined these particles, we saw that they look like they have little vault-sized holes in them. You can see here I've superimposed some vault particles over the holes, and we imagine that those were the vaults trapped in the sol gel. And we could prove that by taking these sol gels apart at pH 9 and releasing the vaults, which were easily released, and they were intact particles with nearly 100% recovery. When we formed the vault sol gels with the MNP packaged enzyme, we increased stability of the enzyme. Here it's shown to be stable to organic solvents like methanol and isopropanol. We saw increased stability to temperature, 30-minute incubation at 45 and 50 degrees, completely inactivated MNP enzyme, but not when it was packaged in the vaults. The enzyme was better even with organic solvents and higher temperature. So the sol gel packaged enzyme worked better than we could have imagined. Moreover, we could reuse these MNP vault silica particles over and over again. Meng was able to do 12 reuses of the particle at catalyzation without a loss of more than 10%, only a 10% loss in activity, even after 30 uses of the particle. This drop after 32 uses is not due to enzyme inactivation, but rather we start to lose the particles themselves because they start cracking when we can centrifuge the particles to reuse them. So to summarize the sol gel work, we demonstrated a neutral templating route to immobilize vaults with very high immobilization efficiency. The sol gels have a high loading capacity. They have negligible leaching. Important, the enzymes immobilized retain their activity with an improved stability and high reusability. So I want to wrap up my talk today with a very brief discussion of vault function. And I'll start by telling you that we don't know what the function of vaults are, but I want to tell you where we are. And all of the questions about vault function usually start with questions about vault knockouts in animal models. And so there's been a lot of vault knockouts in the mouse model. And let me quickly summarize. We first knocked out the TEP1 protein in a mouse model and collaborating with Lee Harrington's group at Amgen Institute. We knocked out the TEP1 protein, and when it was knocked out, the Vault RNA was no longer associated with the Vault, as we suspected, because this is a RNA binding protein. But the mouse was just fine. It had a normal phenotype, and the telomeres of the mouse were also normal. Valerie later collaborated with the same group to knock out the the VPAR protein. And again, when we knocked out the VPAR protein, vaults were still made. The mice were fine, except we later showed a very small increase in induced tumors in this mouse. This was not a significant increase, but we did these experiments because vaults had been suspected to be involved in multi-drug resistance. But since that time, they have not been found to be directly involved in multi-drug resistance. When the RNA was knocked out, we also did a double knockout of TEP1 and VPARP. Again, the mouse was fine. The vaults were still formed. They did not contain either the TEP1, the VPARP protein, or the RNA. They were phenotypically normal. The RNA was still made, but it was unstable. A little while later, a group in the Netherlands, headed up by Eric Wiemer, knocked out the MVP and found normal mice, but they later collaborated with a group at Yale, headed up by Gerald Peer, that showed that these mice had some problems with innate immunity. But we were unable to reproduce these results with our MVP knockout mice. And this group probably had problems as well, because they never published another paper about this knockout. Unlike humans, where there are 3 RNA genes, the mouse has a single MVP gene, and it was recently knocked out by Jonah Vidigal at NIH. And she found that these mice were also pretty much normal. However, there was a slight reduction in platelet counts, perhaps pointing to a role in hematopoiesis. A few years ago, it was shown that the Vault RNAs could be processed into small regulatory RNAs. And recently, Matt Hensey's group at EMBL in Heidelberg discovered that one of the human RNAs binds to and regulates autophagy through binding to the autophagy receptor P62, directly changing P62 function. And I refer you to a recent excellent review on this topic. A few years ago, Valerie produced a triple knockout mouse. All the Vault protein components were knocked out, and the RNA was unstable. But the triple knockout mouse was not seriously affected. It was normal phenotypically, but it had some subtle immune problems, delayed response to certain pathogen infections, and some subtle macrophage abnormalities were seen. So there are many hypotheses for Vault function that have not been ruled out, and they're summarized on this list. My top hypothesis is that vaults have a role in immune responses, but layly there's some new data that suggests that vaults might be involved in intracellular signal transduction. So data is now coming out in that area. But other ideas have some support in the literature, including that they may play a role as storage for amino acids. I just mentioned the role of the RNA in autophagy. Maybe vaults themselves have a role in autophagy. Finally, Vaults may carry out some intracellular transport of cargo. That has not been ruled out, and they may be secreted from cells where they may be signaling. That's also possible. Hopefully, my talk today will inspire one of you to work on vaults and determine vault function. I want to close today with an acknowledgement of the obvious. Everything I described to you today about vaults was gathered over the past 40 years in collaboration with a bunch of talented scientists from all over the globe. In addition, funding for my research has come from multiple sources. I do want to point out 4 people who deserve special attention today. Valerie Kickofer and the labs of Steve Dubonnet, Shali Mendra, and Hong Zhao. They have been key collaborators whose work I mentioned today. Finally, I want to mention that if you want more information about vaults, you can go to the Vault website. And if you want more detailed kind of backstory on the Vault findings and some more basic Vault descriptions, you can visit my YouTube channel where I describe the Vault story in much more detail. I'd like to now return it to Alice. Alice, I hope I stayed on time for you and gave you time for questions.